Good morning, everyone. Welcome to the 5th All-English Golden Brand Research Proposal Presentation Competition. And before we get started, just a quick reminder, please turn off your cell phone or switch to silent mode. Thank you very much. Okay, and this morning we have 6 teams, and we are very honored to invite 3 professors from the College of Medicine to be today's judges. Professor Lin, Professor Wong, okay. Professor Lin Jinru, okay, thank you. And Professor Eddie, Professor Wong Fengru, and Professor Wang Shaoming. Okay, thank you very much. Okay, now before we get started, let's put our hands together and welcome the Dean of the College of Nursing, Dr. Cai Chonghao, to deliver the opening remarks.
Good morning, my dear colleagues. It is my honor to open this Golden Brain competition again. Every year I come to here and have this kind of opening, and wish all of us to have a very good presentation, and also I wish you have a prosperous harvest during this kind of competition. I think this is a very short duration presentation. I think the more difficult for all of us to have a very concise and very precise presentation. But I think all of you have a very good result of your experiment and your study. So I think you must have this kind of potential to have a very precise and a very excellent presentation. And I wish all of you to have a good result from today's competition. Thank you very much.
Thank you, Dean. Okay, now let's invite Professor Lin to explain today's competition rules.
Good morning, Dean Cai, and all the professors and the students. Good morning. Okay, apologize for a little bit delay. We have some technology problems, but I think that should be fine, right? This means that we want you to open your eye widely so you can look more closer on the screen, more closely, or you can look clearly on the screen. And also, this means that to screen, then you will have double portions, double blessings from your presentation. So before I get into the competition rules, I wish you all a very successful presentation. So again, I noticed that some of you look familiar because some of you did quite well on the previous competitions. So maybe some of you know the rule quite well, and some of you probably not. This is your first time to give the presentation. So for this, as Dean Cai just mentioned, it's a short presentation. So it will be a 3-minute oral presentation and plus 2 minutes Q&A. So please, time control is very important. So for those, because time control is also one of the key points in for our giving the score, the gradings. And also, so for those of you, you probably noticed that we have on the screen, we have a time, the timer. So please keep your track on the time. And that's right. For the evaluation and award, we will judge your presentation, and in the next slide, I will give you more detail about how we give the score, how we evaluate your performance. And I think all of you come to join this presentation maybe for the awards, right? The money, okay, the award money. So for the first place, we will give you the first place team. The award is the 10,000 NT$ and a certificate. And the second place is 8,000 NT$ and certificate. The third place is 6,000 NT$. And we also have the Excellence Award, which will be given 3,000 and a certificate, and the Best Popularity Award 2,000 and a certificate. And now let's get to the judge criteria. So on the right-hand side, this part, the theme and the research process and research result. This part comes from your proposal, not written proposal, and the previous proposal that you have been given. And today, your oral presentation and the completeness of the presentation content. Well, oral presentation will account for 30%, and the completeness of presentation content will account for 20% of your total score. So this, I hope, is there any question about this judging criteria? Okay. I hope you are all clear about this. Okay. And today we are very pleased to have 16. And so this is how the schedule about today's presentation competition. And so the first team is the biosensor, and the second team is the NGSAT, and the third team is Tech Talk, and the fourth team is Little Magician. And fifth team is the Penguin, and the sixth team is Hearing Defender. And then we will take a short break for the to give you comments, and then we will announce the award and take a few photos. Okay, and then you can enjoy your lunch. So this is basically today's schedule. Okay, so before we get started, is there any question from our student? Well, if not, then I think we can get started. Okay, so now let's welcome the first team. Right?
Good morning, everyone. Myself, Karya Zainab from the Department of Translational Medicine and New Drug Development. The title of my project is Designing a Fred Probe for Bioanalysis based on a novel AI mechanism. So let's start with the motivation of this project. As our president says, right patient, right treatment. I would like to add one more line: right diagnosis at the right time. So my research project covers the synthesis of the new fluorescent molecule for the development of more efficient imaging material. Our focal point was to design and synthesize the molecule that have the AIE property and which have both theranostic and the bioanalytical property. So what is the AIE molecule? AIE molecule acts as a probe which can be used as a bioimaging, biosensing, and disease biomarker. So the problem with the traditional emitters was that they were planar in shape, and in dilute solution they have the emission, but in concentrated solution their emission got quenched. So the accumulation caused quenching of the fluorescence. So the new emitters are the propeller light shaped. So when in dilute solution they don't have emission, but when bonded to the biological analyte they have the enhanced emission, and accumulative state they also have the enhanced emission. So we designed our synthesis like this. We have the fluorophore tetrafluorine ring as a chromophore. To obtain our final product to 1, we attached a resorcinol group, and to obtain our final product 2, we extended the conjugation with the tetrafluorine amine group. We successfully synthesized both of our compound. To analyze our compound, we did the UV spectroscopy. So our compound second with the extended conjugation shows the UV absorption maxima at 320 nanometer, which shows that the compound second is red shifted, and the compound first is showing the absorption maxima at the 264 nanometer. So we can conclude that our probe has been successfully synthesized, which is a remarkable achievement that opens doors to the limitless possibilities in our research. We have also done the characterization of our product with the UV mass and the on fluorescence to understand the coordination with the metal ion. We did the titration with the europium. To further investigate its fluorescence property, we will do the fluorescence spectroscopy at different concentration in Solomons. Thank you.
Thank you. Can you give us more detail about what's the advantage of the sensor being compared with other type of previous sensor?
Okay, so our sensor is we want to synthesize the biosensor with the AI accumulation induced emission, which have the because the traditional emitters were having the quenching effect in the concentrated solution, but our main motive was to synthesize the a biosensor which have the which don't have this kind of limitation, which will in concentrated solution will have the enhanced emission. Is it clear?
And is there any application that you think your sensor can be used in kind of
So what, as in first slide I mentioned that the AIE molecule can be act as a probe for biosensing, bioimaging, and it can be used also as the probe for the cancer detection.
So basically this is a fluorescent dye, and you mentioned that it's redshifted. So redshifted is in terms of the emission wavelength or the excitation wavelength?
So actually we performed on the UV till it is redshifted in the we can say in the emissions, not the excitation. We have to perform the fluorescence to see that more detailed. When in the detail we have to perform the fluorescence spectroscopy. It's on the way.
Okay, so this is a same excitation wavelength, and then the emission is redshifted. So does that mean the emission color is a different spectrum? So your original color is green, and it's redshifted. The emission wavelength, the spectrum should land either yellow or red.
No, actually we have to, our main motive is to enhance the emission. The redshift means we have the absorption, enhanced absorption. It's not like that the yellow or the redshift.
I see, thank you.
Thank you.
Hello everyone, my name is Jenny Son, and today my topic is the mitochondrial genome study of Alzheimer's disease in Taiwan. And my presentation today will be separated into 3 parts. The first part is why, and the second part is what, and the third is how. So in the first beginning, I want to introduce our motivation. The first is that the rate of Alzheimer's disease is increasing year by year. And some epidemiological surveys also indicate that dementia has become a global health problem. And Alzheimer's disease, we all know that is a predominant form of dementia. It's also increasing in Taiwan. We can see in this chart. The reference is from Taiwan Alzheimer's Disease Association. And it is from about 14 people per thousand in 2021 to increase about 35 people per thousand in 2046. So we all know that through the research about this topic, it's become more and more important. And the second is that the patterns of Alzheimer's disease in Taiwan show distinct difference compared to other countries. In Taiwan, it's lower than Japan, the Europe, and the United States. So we want to know what caused this phenomenon. And our research objective is that we want to determine the correlation between mitochondrial DNA gene biomarkers with Alzheimer's disease in Taiwan. And the second is that we want to find other genomes in mitochondrial DNA specific of Alzheimer's disease patients in Taiwan. And this is our research background. As I mentioned about humanin and smooth, which are 2 important genes on mitochondrial DNA. And there are genes that will produce some proteins, and the small nucleotide polymorphism on those genes will impact the protein levels they produce, and then it will affect the risk of Alzheimer's disease. And this is our research methodology, is that we will use the database from ChinaMedicalUniversity Hospital, and then we will use the MIWAS analysis strategy, and then we will see the results. And what we want to see is our expected outcomes is that we want to find a correlation between the small nucleotide polymorphism of mitochondrial DNA and Alzheimer's disease in Taiwan. And then we will use it as like a biomarkers in some disease and then we will use for the earlier diagnosis of the Alzheimer's disease. And here's my reference and my presentation is end. Thank you for listening.
Alright, thank you for your presentation. So I have a couple questions. Is what alternative approach will you use if you cannot find the mitochondrial DNA change between the normal subject and the AD patient?
And you mean if we conduct the research and then we can't find the difference between the Alzheimer's disease patients and the normal people?
Yes.
I think because we haven't conducted this research now, we only, and our stage is now collecting some information from the database. But this question is very, very important because if there are disadvantage of our research, it may cause some disadvantage of many problems and will cause about a lot of problems. But I think if we can't find the true difference between both of them, I think we can conduct it maybe not the mitochondrial DNA. We can make into the like the chromosome DNA or the other DNA of our cell and find whether there is other genes or other genomes in our body to impact the risk of Alzheimer's disease.
So following your answer, so what specific cells are you investigating in your study?
In our study,
Yes.
the
Depend on your result, Alzheimer's disease. So what are cells are you investigating?
the cells are
So the mitochondrial DNA, the secretion cells, which cells can secrete the mitochondrial DNA, or which mitochondrial DNA are you investigating in the which cells?
what kinds of cells?
Yes, the neuron or other cells.
Actually, because we haven't specific to some kinds of cells with the mitochondrial DNA, we are like the whole, and we want to find the whole and to choose some small or some specific of that.
Good morning, honorable judges and fellow students. I'm honored to be here today to take you on a journey of mending minds. ChinaMed stands with microglia in the fight against depression. So depression, also known as major depressive disorder, is one of the most prevalent psychiatric diseases with multiple negative symptoms, which is believed to be due to systemic inflammation. And current medicines have multiple side effects and long induction periods. Ketamine, however, the NMDAR antagonist, is found to be fast onset. Yet its mechanism is unclear and has multiple side effects as well. So delineating the pharmacodynamics of ketamine can provide us with better solutions against depression. But first, let's talk about what causes depression. The 2K hypothesis is the theory believed by the majority. So usually when the stress hits an adult, we'll feel stressed, definitely, but we can quickly get over it when the stress disappears. However, research has found that lots of depressive patients have encountered stress in their early life and cause inflammation, which can prime their immature microglia, making them susceptible to stress. So even though they can recover from the stress, when the stress hits again in adulthood, the brain inflammation became uncontrollable and caused depression. Coincidentally, the NMDAR pathway is a significant pathway through which microglia cause depression. So we hypothesized that ketamine activate on the NMDAR on microglia to cure the depression. Here's our experimental plot. So we applied an LPS-induced depression model, then we injected ketamine 2 hours before the behavioral test. So these are the behavioral tests we use to evaluate the depressive symptoms, including appetite, anhedonia, and desperation. We also established a microglia depleting model using PLX to inhibit CSF1R. So thereafter, we first found that the depressive symptoms can be relieved by the injection of ketamine, indicating that ketamine has the antidepressant effect. Thereafter, we wanted to know whether these kind of effect can be microglia involving. So as you can see in this figure, ketamine is no longer effective after microglia depletion, which means that ketamine needs microglia to exert its function. So our future plan is to investigate the NMDAR on microglia, see what its role is. We will use this kind of transgenic mice to invalidate our hypothesis. So if the aforementioned hypothesis is clarified, we also believe that the microglia in a specific brain region is responsible for the depression phenotype, such as the anterior cingulate cortex. So if all the question is clarified, we believe there will be a promising future for the pharmaceutical development. For example, maybe you could create some kind of immunotherapy for depression. So in conclusion, we believe through our project, we can make a significant breakthrough in neuroscience field and provide thousands of millions of patients a better choice. Thank you.
Really great presentation. So I know you mentioned that you picked ketamine for a reason, because probably from the article search that you found it's probably the most effective. But I'm still wondering if you ever tried or going to try other open channel NMDA blocker.
Yes, we did. We tried like MK01, the most famous experimental NMDA receptor antagonist, and exactly show the same effects as ketamine did. So this makes us more confident about that NMDA receptor antagonist is going to treat the depression.
Another question is actually two questions. So first, the MK01 and ketamine, which one is more effective in terms of your behavior outcome? And also, there's a third open channel blocker, dangerous dust, PCP.
Yeah.
I know it's probably difficult to get, but
we did have CBP.
Okay,
Yeah,
and also tried it.
We didn't try CBP,
Okay.
but we just did the MK01 and the ketamine, and actually they show the similar effects.
Okay, okay. So does it have to be the open channel blocker, or does it like a regular NMDA receptor antagonist like AP5? Do those
Yeah, we
also
also
work?
did AP5, but I think the recent study has supported the hypothesis that those uncompetitive NMDA receptor is the one who can treat the depression, not other types of the NMDA receptor antagonist.
Okay, thank you.
Did you check the other region in beside your cingulate cortex?
Yeah. So actually, there's another brain region called lateral habenula nucleus, and actually, over the past 1 year, there are 3 Nature publications discussing about this brain region with the ketamine antidepressant effects. So this is what the region we're going to look at. Not only the anterior cingulate cortex, but also the lateral habenula nucleus.
Thank you.
Thank you.
Hello, my name is Xu Yizhen, and my topic is topically applied method modulate social avoidance and dynamic behavior and related neuroinformation. So I want to start with the mechanism under the current clinical psychiatric drugs. They mainly focus on neurotransmitters, which means they mainly focus on neurons. However, years ago, scientists started to notice glia cells and found that they actually take important parts in the pathology of psychiatric disease as well. Like here's an example, microglia. They are the major initial sensors of signal like trauma or infection, and they can fire the secondary inflammatory responses. So what this neuroinflammation will do is to be the amplification of the disease or contributes to the neurotoxicity. So maybe you can guess what I'm going to tell. Like clinically, people focus on neurotransmitter, But as we can know, study also pointed out that neuroinflammation can be a potential target of the therapy. So remember this point. Okay, now going to my project. I use social stress to induce abnormality in minds, just like I mentioned last time. And this is the model I use, and this is my therapy. So as I present last time, we saw social avoidance in RCS mice and anxiety in RCS mice, and it's alleviated by applying method. And we can also see the changes, the protective effect of applying method in RCS mice. And so let's take a small pause here. Remember, we talked about the potential of MS neuroinflammation previously. And an important question of clinical drug development is why this drug is worthy of our attention, why it's different. So this is the interesting part we find this time. We found that by applying method, we can actually alleviate the neuroinflammation. Like these are the expression of pro-inflammatory cytokines, and these are data of anti-inflammatory cytokines. We can also see the changes in chemokines and inflammasome. And this is the protein expression. So here comes to our conclusion. We noticed that neuroinflammation is involved in the pathology of psychiatric disease. And it's surprisingly that we found it's also involved in the mechanism of applying methyl tocopherol. Thank you.
Thank you for your presentation. And we know that the neuroinflammation contributes to neurological disease progression, such as psychiatric disorder, as group said that depression and your anxiety, right? And I have a couple questions because I don't know, I cannot catch all your result. The first one, why did you check the NAOA and NAOB?
Because the present clinical drug focus on focus on this enzyme. So we want to show that it's do have changes.
Because it have changes, so you check that. However, your rationale, why did you check the NAOA and NAOB?
Because, like, after we found that it have protective effect.
Maybe you can say that because you use the RNA-seq or you use the other study to check the NAOA and the NAOB can be checked. So you focus on these two genes. However, you did not say that your rationale. Do you know what I mean?
Can you?
Because you just say the NAOA and NAOB can be changed in your study. However, you don't talk to me this coming from maybe RNA-seq, RNA sequence, or RNA.
Okay, and this is what we are going to check. We have two hypotheses. Now, first one is that methyl pass through the skin and go into the blood circulation and act on the brain cells. And the second one is it strikingly act on the receptor on our skin. So we are going to check it by block the receptor related.
Ladies and gentlemen, my name is Ke Yi-Tong, and my topic is circadian clock of tumor to improve cancer treatment. The core goal of clinical cancer research is to find out ways to increase the sensitivity of cancer cells to conventional anti-cancer regimens. This is far more practical than constantly developing new drugs. Studies have shown that chemotherapy can enhance the response of tumors to anti-cancer drugs. Therefore, this study aims at further investigate whether Tagore circadian clock of tumor can improve the cancer treatment. Our body has a natural rhythm called circadian, which is regulated by eating and sleeping behavior, and it will control the metabolism and various functions in our body. When this rhythm is disrupted, it will cause various diseases in our body, which will end up leading to the increasing risk of cancer. Moreover, in clinic, chemotherapy can enhance the response of tumor to drugs without additional medication, but only by administering the drugs in specific times. So, why chemotherapy is effective and how to use circadian to improve the cancer treatment are both important direction for future development. We all know that circadian is critical to maintain the glucose metabolism in our body. In addition, previous studies suggest that in tumor, the glycolysis will reprogram to lead to increasing resistance of the conventional therapy. So, according to all the studies, suggest that well circadian will control glycolysis, which make the tumor sensitive to anti-cancer drug. In contrast, increasing resistance to anti-cancer drug may be due to the aberrant circadian or the glycolysis reprogramming. To investigate the mechanism behind aberrant glycolysis and aberrant circadian, I will use the pancreatic cancer cell lines and oral cancer cell lines. Treat the 5-fluorouracil and cisplatin to treat them. After using the dexamethasone to synchronize all the cells and induce their circadian. Subsequently, I will use the RT-PCR and NTS assay are performed to predict the expression, time-dependent expression of circadian and glycolysis gene. Also, to analyze the drug sensitivity upon the different time treatment upon different times. In conclusion, we believe that chemotherapy, understanding the mechanism behind aberrant chemotherapy, aberrant circadian and glycolysis will help us rescue and enhance the sensitivity of anti-cancer drug. Thank you for listening.
All right, thank you for your presentation. It's very interesting study. And in physiology, does the circadian affect cancer progression first, or does cancer disrupt circadian to promote cancer progression? So which one is the first?
And do you mean circadian? Can you say the question again?
Circadian. Does the circadian affect cancer progression first, or the cancer disrupt circadian rising to promote cancer progression?
Based on the paper I have seen, I think maybe it's the cancer disrupt the circadian first.
Oh, all right, so the cancer disrupt the circadian to promote cancer progression, right?
Yeah.
So, but in your study, you synchronize the cell into the at the same time and then treat the anti-cancer drug. So this one is the circadian affect the cancer progression. Do you know what I mean?
Yeah. I'm thinking about my study is about to see whether just grouping the cells to divided them for two groups. One is circadian related, and the other is circadian non-related. So I'll grouping the cells, and for the further experiment, I can use the RNA sequence to see if there are differences, RNA or genes between those two groups of cells.
All right, thank you so much.
Good morning, everyone. My name is Zhang Yushen with Team Carry Defender. Today we are talking about a computational analysis of workplace ear loss selection for preventing occupational nurse-induced hearing loss. If you work in a hospital, you can listen this kind of sound with a prevalence about 18% hearing loss. And the contraction size is about 23%. And the warning size is about 27%. It sounds very annoying, right? However, we have to stay there for a long time. Based on our previous paper, we used binary element analysis to predict the hearing loss in a specific workplace. Here, our objective is to deploy our core algorithm through a mobile app, which will record workplace noise and analyze it. FEA can help us understand the damage on the basal membrane. After that, hearing can be protected by our tailor-made earplugs. Our earplugs consist of 3 parts: earplug cap, supporting structure, and noise filters. We will focus on designing a noise filter to meet the needs of various workplaces with different noise patterns. Our preliminary data showing different materials has different noise attenuation abilities across the sound frequency. Traditional foam material is the least effective and not useful compared to others. It should be replaced by other materials. Our clinical study showing above 65 decibel, which is the hearing loss threshold, because the hospital staff had a risk of hearing loss. And we measure the hospital clinical laboratory noise exposure. It's about 74 decibel. So let's take the hospital scenario as an example. This is our business model. First, clients just need to use a mobile app to record noise and upload it to our database. It's showing 2000 Hz is a high sound pressure level in the hospital. Next, after calculating by our core FEA algorithm, showing that 2000 Hz is the most damaged frequency to our to our ears. Then back to the client's app. Our algorithm suggests that silk and collagen HA are the best choice of the noise filter materials due to their ability to filter the harmful frequencies and still allow the wide long frequency to be heard. So we will recommend to the hospital staff. Finally, the factory will produce and send it back to a customer. This is our business model. Our product, high-tech and eco-friendly. Thank you for listening.
Thank you for your presentation. So, I want to just to make clear that so the earplug it filters the frequency, the sound frequency about 2000 hertz, right? Is that right? According to your thing?
According the different materials, so we can attenuate like different frequencies. And from this scenario that we measure the noise in the hospital, and the most damaged frequency will be like 2000 hertz. So, yeah.
But I think for our human ear, the most hearable frequency will be around like 1000 to 2000. Okay, so does that mean you also filter most of the, maybe some of the necessary?
Yeah, that's a problem that we think of. And because like talking is like about 500 to 4000 hertz. And we specifically will attenuate like 200 hertz because it do more damage to our ear. So like we can still talk to each other, and on the other frequencies it won't be attenuated so much, so we can still hear others.
And also like so currently in the market there are so many earplugs or this kind of so what's the advantage of your product?
Okay. So these are the 3 earplugs that we now are available on the market. And so from traditional foam earplugs, it cannot very specifically block out some very specific frequencies. And for earmuffs, they are more heavier, and the comfort is the main issue. And it may be more humid in a space of the ear. And even for wearers they may not get a good seal. And for titanium earplugs, as you can see, it is obviously just too expensive. And for our hearing defenders, we can provide a more efficient, more durable earplugs for everyone, and not only benefit the users, but also benefit the whole environment. Yeah, thank you.
We need your coordinations to vote for the best popularity team. So please take your cell phone and log into the Slido. Oh, okay, you can scan. Okay, you don't need to log, you can scan.
So each person can select 3 teams that you like the best. Okay, anyone needs more time or everyone has finished? Select. If you need more time, please raise your hand. Okay, I see one hand. Okay, I'll give you 30 more seconds. Is that okay? Okay? Anyone need more time? No? Okay. Okay, so we have the result. So the first, the most popular teams are Hearing Defender, Little Magicant, and NGSAT. Okay, congratulations. Okay, so now we will take a 15-minute break, and when we come back, we will have the award ceremony. Thank you very much.
Right, so I think we can, now is a very important moment, right? This is a moment you are all waiting for. But before we announce today's award, the award, so we, the judges, we just talk about that. I think today, even though we just have 6 groups, but we think all of you did a great job. You all did your high quality research or high quality presentation. So I think you should all give yourself a big applause. And one of the suggestions that we agree, all judges we agree is that because I know you spend lots of time doing your research, but we suggest that you mention or put your advisor's name in the slide, in the very first slide, because you need to honor your advisor or honor your team members, or maybe mention which lab you are from. So let us know that who your advisor is, and so that will give us a clear idea. Because this nowadays the research comes from teamwork, and also you need your advisors give you some suggestion or guidance. So this would be a good time to honor your advisor. So that's a suggestion that we want to give all of you. Okay, so besides that, I think, as I mentioned, I think you all did a great job. But still, we need to come up with the place 1 and place 2 and all different so the award. So now let's go with the best popularity award. So the best popularity award, as you just saw, you can previously before the break, you can see on the screen. So the best popularity award goes to Group 2 team, MGSAT, and also the Group 4 Little Mexican, and also the Group 6 Hearing Defenders. Please come to the front. Congratulations. Okay, congratulations.
Little bit more. Professor.
Team 1 Biosensor, and the Team 2 MGSAT, and the Team 4 Little Mexican. Okay. Yes, so Professor, welcome Professor Wong to give the award. Please come to the front. Congratulations. Yeah, as I mentioned, you all did a great job. And the third place goes to Team 5 Penguin. Team 5 Penguin. Congratulations. So then, let's welcome Team Thai. The second place goes to Team 6, Hearing Defenders. Team 6, congratulations.
Let's go. No, I'm not going. Where will you go? Jimmy.
And the first place goes to Team 3 Tech Call. Congratulations.